In the previous video, we discussed about the structure of the cell membrane and the integral proteins and peripheral proteins and the structure of the phospholipids. So today we'll discuss about transport by cell membrane, how one molecule can transport from the extracellular fluid to intercellular fluid or from intercellular fluid to extracellular fluid. At first, we will discuss about simple diffusion. The simple diffusion will occur only for the gases like oxygen and carbon dioxide and the lipid soluble molecule and small molecular mass. For example, if this is the cell membrane, then the oxygen concentration inside the cell is less due to the process and metabolic process of the cell, but the oxygen in the blood vascular system is greater than the inside of the cell. So oxygen will conduct from outside to the inside of the cell, but the amount of carbon dioxide or concentration of carbon dioxide is very high inside the cell due to metabolic process, but carbon dioxide is very low amount outside the cell. So the carbon dioxide go out to the cell. This occurs by simple diffusion. It does not need any energy or it does not need any ATP for this process. This diffusion process are dependent on some concentration gradient or surface area or solubility or thickness or molecular weight of substance. We will look after how these are connected with the rate of diffusion. The rate of diffusion is directly proportional to concentration gradient. If the amount of oxygen outside the cell is higher, then more amount of oxygen can enter into the cell. If more amount of carbon dioxide is inside the cell, then more amount of carbon dioxide can go out of the cell. Surface area. If the surface area is this much, then only little amount of O2 can enter. But if the surface area extended to this much, then more amount of oxygen can enter into the cell. This is dependent on surface area. So if surface area increases, then diffusion also increases. If concentration gradient increases, then diffusion also increases. Directly proportional to solubility. More amount of solubility of oxygen is in the blood, then more amount of oxygen can enter into the cell. More amount of carbon dioxide solubility inside the cell, then more amount of carbon dioxide can transport outside of the cell, and is inversely proportional to thickness. If thickness increases, then the diffusion decreases. If the thickness of the membrane this much, then there is only little path to travel for oxygen or carbon dioxide. But if thickness increases to this much, then it has to travel more far, so the amount decreases. So if thickness increases, then amount of diffusion decreases. It is inversely proportional to molecular weight of substance. If the molecular weight is more, then no substance can enter into the cell with high molecular weight, like thyrox T3 or T4 can enter into the cell by simple diffusion, but insulin cannot enter into the cell through simple diffusion. So it also dependent on molecular weight of the substance. Then at second we will look up facilitated diffusion. Facilitated diffusion also don't need any external energy or ATP. It's important. Diffusion also don't need any ATP, and facilitated diffusion also don't need any ATP. It is just facilitated by a channels, or it is a protein. We look in previous video that it is sometimes water filled channels, and these are made up of proteins, polypeptide chains, which are made up of amino acids. Here these channels are three types. One is leaky channel, another one is voltage gated, and another one is ligand gated. In leaky channel there is no barrier on outside or inside. From extracellular fluid it will come directly to intercellular fluid, directly depends upon the concentration gradient. So it is always open. It is the cell membrane, and from extracellular fluid to intercellular fluid or intercellular fluid to extracellular fluid. Like this, potassium travels through the leaky channel of the cell membrane. We will discuss in resting membrane potential elaborately. In voltage gated channel, the channels have gates either outside or inside or both sides. These are only open at certain voltage, like sodium channel only open, let be example, in 60 millivolt. So it will open only in 60 millivolt but not in 90 millivolt. Minus 60 volt minus 90 millivolt. So these are voltage gated channels. Ligand gated channels, like this. Here one ligand will attach, any hormone or any substance will attach, then this channel will open for any substance. These are ligand gated channels. Then we will travel forward to active transport. What is active transport? Active transport need ATP. So these are called active transport. At first we will see about primary active transport. What is primary active transport? In primary active transport, ATP is converted into ATP to give one phosphate as energy molecule to carry out the process. Here 2K plus is come into the cell and 3 Na plus are taken out of the cell. This is called Na plus sodium potassium ATPase. These conduct primary transport and ATP is converted to ADP at this site. So this is primary transport. Like this, calcium also came out of the endoplasmic reticulum through this process. Here ATP is converted into ATP at here only. So it is primary active transport. But if here, if this is an gastrointestinal cell, here in small intestine, sodium glucose sympod will work to take glucose the GIT from the GIT to the cell. Here sodium, we all know that sodium concentration in the cell is very low and outside is very high. So sodium comes into the cell. With the sodium, the glucose also come into the cell. This is called sympod because two molecules are coming into the cell simultaneously. So here no ATP is used directly, but to decrease the sodium concentration of the cell, we have to activate this process, this activate, so that the sodium can go out of the cell. So here ATP is not work directly, but here ATP are used indirectly. So these are secondary active transport. And in another case, the sodium is outside connected to one protein and the hydrogen proton connected to another inside. When sodium comes inside, the hydrogen proton go outside. This is also one process in which hydrogen go outside of the cell. This is called antipode because both are working in opposite way. So if two molecules are attached and they both simultaneously coming to the cell, they are called sympod, and if they are going in opposite, they are called antipode. Carrier mediated transport has specificity. Here we saw the carrier mediated, here carrier mediated, here also carrier mediated. We saw some specificity, like they have only specific characters. First specificity is stereospecificity. Like this, here we know sodium glucose transporter. Here one protein, here sodium, here glucose. Here only glucose can bind to this sympod, not other molecule can bind to this. This is called stereospecificity. Next one, phenomenon of saturation. If there is, it is the wall of cell membrane, and here six symport are present. 6 symport sodium glucose symport are 6 sodium glucose symport are present here. So if there is here 6 glucose, then it will connect it to 6. If there are 5, then 5. But if there is 7 or 70, they cannot go into the cell by the 6 transporters simultaneously. So this becomes saturated. These have only a saturation point till which it can work. Then if you increase the concentration of glucose, it cannot work as higher. Like in this graph, the velocity of the symport into the cell, and here the concentration of glucose we take. At some extent, if we increase the glucose concentration, the velocity increases, and after some time it will remain steady. So this is called saturation and competition. Sometimes here we will take an example, this input only. We have sodium and glucose. Sometimes glucagon. Sometimes glucagons are connected to this glucose receptor, and it creates a competition for glucose to import inside to the cell. This is called competition. Then we will go for vesicular transportation. How the vesicular means here is one cell, let's here is one cell, and here one vesicle created, and it go into the cell. How this works we will understand here. Vesicular transportation are two types, endocytosis and exocytosis. Endocytosis means some things go into the cell, and exocytosis means some things go out of the cell. And endocytosis have three parts. First, pinocytosis, phagocytosis, and receptor mediated endocytosis. And exocytosis are hormone release and neurotransmitter. First, endocytosis. If there is a cell here, inside there is intracellular fluid and outside is full of extracellular fluid. So sometimes here small, due to the fluidity of this cell membrane, small vesicle will approach and it will engulf some water with some molecule from the extracellular fluid, and it go into the cell. This is also called cell drinking. Cell drinking. In pinocytosis it occurs. In phagocytosis mainly occurs in WBC. One microbe is here. We have to capture it and go into the vesicle into the cell. So the cell will perform pseudopodia. Then it pull it into the cell. The pulling of the cell or pseudopodia mainly helped by one molecule called actin. We also read it in previous video as a cellular structure or cytoskeleton. Then receptor mediated endocytosis. If there is a cell, let it be one hepatic cell or liver cell, hepatocyte. Then at first small depression will created here, and here the receptors present for the LDL. Then LDL will connect here, and some molecule will present here, some molecule present will pull the cell membrane inside. This molecule are called catherins. These molecules are called catherins. This molecule pull the membrane inside, and it will create a vacuole, or we will call vesicle, and here the both receptors and LDL are present. Then another vesicle will dissolve with this, and then receptor will then become to the cell membrane again, and LDL will be deposited inside the hepatocyte. So here the receptors are used. So these are called receptors mediated endocytosis. Then Here hormone release. It is so simple because hormone release is just like here beta cell of pancreas, then here insulin is secreted and insulin will go near to the cell membrane, then cell membrane will create a structure like this and then insulin will go out of the cell and this will cell membrane will repair itself again. Then neurotransmitter. In case of neurotransmitter, there is a neuron here and another neuron here. Let it be acetylcholines are secreted from here from the actions of the cell membrane of a neuron and it will come into the cell through neural junctions. Neuro, so it is called neurotransmitter release. Let it be example as acetylcholine. So we'll discuss about how these substances are moved through the cell membrane inside the cell. Let's review the topic again from the first. Transport of the cell membrane through simple diffusion. Then rate of diffusion depends upon directly through concentration gradient, directly to surface area, directly to solubility, indirectly to thickness, indirectly to molecular weight of substance. Then facilitated diffusion don't need any ATP, it's just facilitated by a one protein. It may be leaky, which have no gate, always open, may be voltage gated, which gates are opened by voltage, or ligand gated, which gates are opened by one ligand attached to the space for ligand. Then active transport, it may be primary active transport, which use ATP at the site only directly. It is called one first degree or primary active transport. This may be use of calcium transporter in endoplasmic reticulum, or common example is sodium potassium ATPase. And secondary active transport uses ATP indirectly like sodium glucose transporter and sodium hydrogen transporter. If two hands of this transporter are outside of the cell or inside of the cell and they move simultaneously, it's called symport. And if they move opposite to the extracellular intracellular fluid, they are called antiport. Then character-mediated transport or specificity. Specificity means only one molecule can attach to this site, other molecule cannot. Saturation. If they have limited number of receptors or limited number of symport, then only that amount of glucose only can enter into the cell. After some time, it will keeps a straight line structure in the graph that it cannot increase the velocity of the endocytosis. Competition. If glucagon is like glucose and it creates a competition for the glucose to bind into the sodium glucose symport. And vesicular transportation, vesicular endocytosis and exocytosis. In endocytosis it is pinocytosis, phagocytosis and receptor mediated endocytosis. And in exocytosis it is hormone release and neurotransmitter. And in next class we will discuss about resting membrane potential, how it is created and how it works in our cell. Next video we'll discuss about resting membrane potential. Thank you.